Good day, everybody. This is Chris back again with OEM 201, Human Pathophysiology for Paramedic Prerequisites. It'll be spring 2017. We're going to be continuing our discussion of immunology. So what I'd like to do today is I'd like to dive deep into antibodies, how antibodies are produced. We know that antibodies are produced by plasma cells. These are B cells that have been converted into their active form, and we typically call that active or antibody producing form of B cells plasma cells. And then when they quit producing antibodies after the infection or the insult has been dealt with, they turn into their less active or their dormant form known as memory cells. And then those memory cells can be turned back into plasma cells if the patient or the person is re-insulted or comes into contact with the specific pathogen that has that antigen on it. That can elicit that response. And of course, a very important part of converting a dormant B cell or memory cell back into a plasma cell involves the helper T cells. Okay, so let's just kind of look at the bigger picture going on here. So again, antibodies are also known as immunoglobulins, and we often see this abbreviated by Ig. So when I start talking about IgD or IgA, that's immunoglobulin D or immunoglobulin A. That's a classification system for groups of immunoglobulins or antibodies. But just remember, whenever you hear the term immunoglobulin, really what we're talking about are antibodies. It's just a synonym for that. Okay, so how do antibodies work? Well, antibodies are able to attach to the antigen on a bacteria, some cell, pathogen, even viruses, or even virus-infected cells can produce certain antigens. And in fact, it is the antigen that is used to produce the antibody in the first place, and we'll talk about that a little further down. So what happens is the antibody binds to the antigen, and we're talking proteins here. Typically, for something to be antigenic, it has to be large enough and complex enough to elicit an immune response, and that typically is through the agency of a protein, because as we know, or as we learned about earlier in chemistry, biochemistry, proteins have a fairly significant detailed structure. They have, of course, the primary, the secondary, the tertiary, and quaternary structure, and there's lots of folding that goes into these molecules. So they're actually very useful when it comes to identifying self and non-self because they're so nuanced and complex. Okay, so what happens is the antibody binds to the antigen and forms what's known as an antigen-antibody complex. And this can result in several things occurring. The first thing that can occur is this can go on to activate the complement pathway. And we haven't talked about the complement pathway in any significant detail yet. And I actually may do a video on the complement pathway. But the complement pathway is a very important part of modulating the or the inflammatory response. So complement pathway can help exaggerate or enhance the inflammatory response, and the complement pathway can also help produce proteins, as we talked about in the last set of videos, produce proteins that are very good at puncturing bacteria and hopefully in helping to fight or kill off bacteria. Another thing that can happen is cell-bound antigens, particularly. So if you have a bacteria cell or perhaps a virus-infected body cell, when those antibodies begin binding to the antigens on those cells, it can cause those cells to clump together, or what we call agglutinate. And when agglutination occurs, you kind of pull the bacteria, the virus, the pathogens, you kind of pull them together and you hold them together, and that allows for the immune system, for the other natural killer T cells, phagocytic cells, and other cells to get to that clump a lot easier and to attack it. So it makes it much easier than trying to attack individual pathogens. You can agglutinate them, pull them together, and make a much more efficient immune response. The antigens also are very good at causing antibodies are very good at causing precipitation, and whenever something precipitates, that means to draw it out of a solution. And so normally when you have pathogens, they're floating around, they're kind of dissolved in our blood, and if you could pull them out of our blood, and this is actually part of what's going on with agglutination, if you pull them out of the medium, again, it makes it much easier for the immune system to respond and to get immune cells to those pathogens and to neutralize them. In addition to that, antibodies are very good at direct neutralization of certain pathogens, toxins, poisons, and or venoms. And when I talk about poisonous versus venomous, particularly in response to animals, there is actually a difference between a poison and a venom. And the fundamental difference has to do with the delivery mechanism. This is somewhat of an aside to the specific lecture at hand, but when I say that something is venomous, when I say that an animal is venomous, or I'm talking about a venom, what makes something venomous is the delivery mechanism. There has to be an evolved mechanism for delivering that specific toxic substance. So for example, with a snake, the delivery mechanism would be the fangs, right? The fangs are able to inject that venom into the victim. However, if I'm talking about a frog, say a poison dart frog, where it gets agitated and it secretes a toxic substance out of its skin, there really isn't any specific delivery mechanism for that. It just secretes it, and if you happen to come in contact with it, you may have problems. And so we would call an animal like that poisonous, whereas an animal that has evolved an elaborate delivery mechanism, we call that venomous. So that's really the difference between poisons and venoms. Okay, back to the lecture at hand. So let's just talk about the general process behind antibody production. So here you have some sort of pathogen. This is, say, a bacteria, and this bacteria has proteins on its surface that are antigenic. That is, that the immune system can use them to create antibodies. So what happens is the phagocytic cells, okay, these are our kind of our frontline defenders, right? These are the macrophage, the neutrophils, the dendritic cells in our tissues, right? These are the primary defenders when the first line, when our innate immunity fails. You have a breach in your skin, for example. These are the frontline soldiers that are there, okay, the phagocytic cells. So they phagocytize these pathogens, and then what they do is they pull the antigens away and they present the antigens on their surfaces, and these antigens then get presented to the B lymphocytes. Remember, the B lymphocytes are a part of our acquired immunity, and these are the lymphocytes that can go on to become plasma cells. So what happens is the antigen presenting cells, the APCs, that's why we call them APCs, is they attack the bacteria, they phagocytize it in this case, and kill the bacteria, and then repurpose the antigens. They're like, hey, I can use these to identify more of these bacteria. So then they phagocytize it, they present the antigens to the B cells. The B cells then take the antigens and incorporate parts of the antigens into what's known as the antibody. And then the B cells can then secrete or release the antibodies out into the blood plasma. Those antibodies, because part of the antigen was used in them, are kind of like a key, if you want, and they will only fit the specific antigen that was on the original bacteria. So that allows them, if they bump into the bacteria, they can bump and then they can attach to the antigen there, and then potentially cause all of these things to happen here. Now, it need not necessarily be a bacteria. This could also be a virus. It could be a fungus. It could be a virus-infected cell. It could be an improperly functioning body cell, for that matter. Or it could be a normally functioning body cell, and antibodies could be more or less accidentally created as well. And we call that autoimmunity, where the immune system attacks its own healthy cells and tissues and receptors as well. And we'll talk about that in a little more detail a little later on. Okay, so that's just kind of the general idea behind behind what antibodies are and how they're produced. Okay, so what I want to do now is I want to talk a little more detail about antibody structure and function. So in general, many types of antibodies have a kind of a Y-shaped structure to them. Not all of them, but kind of the fundamental way of looking at antibodies, it kind of has this Y type of structure, and certain antibodies actually have multiple Y-type structures arranged in a more complicated structure, but fundamentally we're still talking about this structure. So what we have here, we have two general parts of the antibody. You have this longer part down here. This is known as the heavy chain, or the Fc domain, this part right down here. That stands for fragment crystallization, or the constant domain. So this is a fairly constant, not a whole lot changes, and you see these little C's and these little N's here. Remember when we talk about making proteins, the fundamental way that we make proteins, and antibodies are a type of protein. Remember when we talked about taking amino acids and combining amino acids to make peptides, and then make polypeptides, and eventually the side chains on the amino acids begin interacting. That causes the polypeptide to fold into its conformational shape, and that makes the protein. Well, what happens is, remember, you have a nitrogen-containing group of one amino acid bonding with the carboxylic acid group of another one, right? And so what happens is you get the C-terminus, right, the carboxylic part of one, and the N-terminus, the nitrogen-containing part of another. And so that's what you see here, is you've got the C-terminus and the N-terminus. And I just kind of threw that in for the sake of completion. But here in the heavy chains, or the Fc domain, there are some minor immunological things that happen, but a lot of what an antibody does is actually in this distal part here, this Y part here, and this part is known as the Fab domain, or the fragment antigen binding domain, and there's much more variability in this area here, depending on the antigen that this antibody is binding to. And so you have lots of receptors and lots of different protein fragments that can help this antibody bind. So this is actually where the antibody binds to the antigen, right in here in the Fab domain. And then you can see that you have two what are called heavy chains, okay, and then two light chains here. And what happens is these heavy and light chains are bonded together primarily through disulfide bridges and other non-covalent bonds, or what we call the intermolecular forces. So we talked about some of these in biochemistry: hydrogen bonds, induced dipole interactions, London dispersion forces, et cetera, et cetera. The disulfide bonds involve sulfur atoms bridging together, two sulfur atoms, disulfide, how about that? And that's actually a really important part of what binds the heavy chains together here, okay? So that's typically what we have going on. You've got lots of your binding receptors, and your actual antigen binding sites tend to be right here at the end of the Fab domain, or the fragment antigen binding domain. Okay, so let's now talk about general classes of antibodies. So you have 5 general classes. You have IgG, IgM, IgA, IgE, and IgD. And remember, these IG stands for immunoglobulin, and immunoglobulin is just an antibody. So I'm going to start, not in alphabetical order, but I'm going to start off with the most abundant antibody in the body, and this is IgG. It is the most prevalent throughout our body, and what's important about this is it can cross the placental barrier, and so what this can do is newborns can have some of mom's antibodies in it and provide it with some temporary immunity. And so IgG antibodies, not only are they the most abundant, and they're very common in attacking viruses, toxins, and different poisons, as well as activating the complement pathway. They're very important because they can cross the placental barrier and they can provide some temporary immunity to the newborn in the first 6 months to about a year of life. Okay, IgM is the second class that I want to talk about, and this is actually the largest antibody, and this does not have this typical Y-shaped structure here, but rather imagine 5 of these Y type of antibodies bound together in kind of a circular structure, so something that looks like this. And that's what an IgM antibody would look like. And they're all joined at the Fc domain, or the constant domain, the fragment crystallization there. They're by their tails, for lack of better words. The IgM antibodies are very important when it comes to agglutination. So when it comes to pulling, right, when it comes to pulling things together, agglutinating them, and we see IgM antibodies play a very important role during the early phases of an infection. Okay, IgA is next, immunoglobulin A. These are primarily found along the mucosal surfaces in saliva, tears, and in your blood as well. These are very important when it comes to protecting body surfaces. So anywhere you have the body or a body cavity interfacing with the external environment. So think about your respiratory mucosa, your gastrointestinal mucosa, your eyes, right? Your eyes, for example. This is very important in protecting those surfaces. And an important part of IgA is it is found in breast milk, and it does seem that there is some evidence that suggests that breastfeeding may also be able to provide some degree of immunity to the newborn through the IgA antibodies that infant receives. There's a lot of discussion about how important this is, and I won't be able to dive into that in any detail in this course. Okay, the next antibody or immunoglobulin I want to talk about is IgE, and this is also like IgA. It's found in many mucosal surfaces. We find this in the lungs, in mucous membranes, in the skin as well. These are very good at reacting to foreign substances, not necessarily pathogens. Okay, and these are very important when we talk about allergies and anaphylaxis. Okay, so people that have allergic reactions and anaphylactic reactions, and anaphylaxis is just a highly exaggerated allergic reaction. IgE antibodies play a very important role in that. And then IgD antibodies are not particularly well understood, so there's not a whole lot that I can say about them in this particular course. Okay, before we move on to the next, the third and final act of this lecture, if you will, I do want to talk about the importance of antibodies when it comes to toxicology in cancer. So what we can actually do is we can expose animals to certain antigens. And these animals, that will elicit an immune response in these animals, and then these animals will go on to create antibodies. We can then take those antibodies, and we can concentrate them and turn them into medications. And this is where certain, or many types of antivenoms, this is how many of our antivenoms are created. And one of the most important antivenoms that I want you guys to be aware of, particularly where we live, is something known as crofab, right? Crofab. And basically what is done, CRO stands for crotalids, okay, it's a type of snake, and then FAB stands for fragment antigen binding, right? So it has something to do with this part of the antibody. So what we do is, I believe this is horses. I believe this is done in horses. Horses are exposed to different types of snake venom. In this particular instance, the crotalids, okay, the pit vipers, so rattlesnakes, for example. And there are, I believe, 5 specific types of snakes. Let's see, I believe it's the sidewinder, the cottonmouth, the western, is it the western diamondback, and is it the Pacific rattlesnake? I believe those specific snakes, those venoms, horses are exposed to those venoms, the horses produce antibodies. We then extract those antibodies, concentrate those antibodies, and we add enzymes to those antibodies that cleave the FC domain, that cleave the constant domain away from the Fab domain. And so all we have is a concentration of the Fab domain of that antibody, and that's why we actually call it CRO-Fab, right? It's not the full antibody, it is just the antigen binding domain of that antibody. So CRO-Fab, very important antivenom for snake bites, particularly down here in the Southwest. There are some newer antivenoms that have hit the market. Antivip is, I believe it's Antivip is one of the newer ones that may be playing a more important role. If it is as effective as CRO-Fab and the costs can be kept down enough to compete on the market. Another important role in toxicology are certain drugs. One of the most common drugs that has an antibody is digoxin. So what we can do is, if somebody has digoxin toxicity, digoxin overdose, of course digoxin is a cardiac glycoside, it's very commonly used to treat heart failure, atrial fibrillation, and other rapid atrial dysrhythmias in some cases. If someone has overdosed or has digoxin toxicity, we can give them something called Digibind, which is an antibody that attaches to the digoxin molecule and causes precipitation and agglutination of that molecule, pulls that molecule out of the blood plasma, and then that molecule is no longer able to attach to receptors in myocardial tissue, and you can effectively treat the toxicity that way. Another really interesting area is in cancer, and an example of antibodies' important role in cancer, something known as the monoclonal antibodies, or MCAs. And monoclonal antibodies are basically antibodies that come from one specific type of cell. So as you might imagine, their Fab domain is going to be very specific for a certain receptor. And this is important because certain types of cancer cells have specific antigens on their surface, express specific antigens. And if you can create an antibody that will attach to those specific receptors, you can target that specific cancer cell and leave the other cells alone. And this will allow you to make a drug that is highly effective but is not as toxic to the other cells. Now, that's a problem with a lot of these chemotherapeutic agents for cancer, is that they are very toxic to any kind of cell that may be dividing because they alkylate DNA. And so they're not as specific as you'd like them to be, and you get lots of side effects, right? You get anywhere you have cells that are dividing in your body, you get lots of side. That's why cancer patients are so immunocompromised, because you have lots of cell division occurring in your immune system, right? You've got lots of stuff going on. And so when you give somebody a chemotherapeutic cancer agent, you're going to suppress their immune system, because those drugs will hit the immune system. They will cause problems in other areas of your body where you have rapidly dividing cells, such as hair follicles and your gastrointestinal tract, your mucosa. And this is where a lot of the side effects of cancer therapy come from. Well, what we can do is we can create these antibodies that are very specific to these cancer cells, and this has been very effective in some forms of cancer. There is one specific agent among many. I'll talk about this one for no particular reason other than just to give you an example, and it's something known as Rituxan. And Rituxan is kind of become a mainstay therapy for a certain type of non-Hodgkin's lymphoma. The lymphoma is a cancer of the lymphatic system. And prior to the introduction of these monoclonal antibodies like Rituxan, something like non-Hodgkin's lymphoma was much more difficult to treat and had relatively high morbidity mortality rates associated with it. And we've actually been able to make a significant impact by developing and using these monoclonal antibodies. Okay, so those are just three examples of the important role that antibodies have outside of the immune system as actual therapeutics in the healthcare setting. Okay, finally I want to end on in this lecture talking about the different types of hypersensitivity reactions, and these make really great test questions. So when we talk about hypersensitivity reactions, we have 5 general types, and these are hypersensitivity reactions that are immune mediated and typically involve antibodies, antibody mediated hypersensitivities. So type 1 primarily involves IgE or immunoglobulin E, and this is where your seasonal allergies come from and anaphylactic reactions. Okay, these are mediated primarily through the immunoglobulin E specific types of immunoglobulin E. I should say that there is a word that you will hear often used to refer to somebody who is allergic or has had a hypersensitivity reaction that is an antibody type of response, and that is known as atopic. So whenever you hear somebody say that a patient or a person is atopic, I just associate that with allergy. Atopic is a type of allergy, a type of hypersensitivity reaction. Okay, moving right along. Type 2 hypersensitivity reactions are also antibody mediated, and the big thing that we need to know here is this leads to cytotoxicity, right? Cell toxicity. So people that have type 2 hypersensitivity reactions have direct cell destruction, okay? And these are really important when it comes to transfusion reactions, for example. ABO transfusion reactions, very important part of that. So when somebody receives a blood transfusion and they have like a hemolytic reaction where you have lots of breakdown of red blood cells, this is a type 2 reaction. Antibodies attach to the red blood cells, cause them to undergo hemolysis, break down, and of course that can be very problematic. Hemolytic newborn diseases, another good example, and this is something that we can see where, of course, you guys are familiar, or you should be familiar with the A, B, O, and RH, right? So red blood cells can have the A antigen, they can have the B antigen, or they can have both, and we call that AB. And then O, type O blood, is blood that has neither A or B antigens, and then the RH factor is the D antigen. Well, mothers who are RH negative, okay, so if you have an RH negative mother, and that mother gives birth to an RH positive infant. So what can happen is the first time you get pregnant, this is generally not a big problem. Okay, and there is this concept known as sensitization. I believe I talked about in the last video, where typically the very first time you're exposed to something, you stand a good chance of not developing an immune response because your body has to identify that as an antigen. It has to create memory cells, and by the time all that occurs, the situation may very well have resolved. So typically the first time that you are exposed, you are sensitized, and then subsequent exposures are where you can develop allergic reactions and even anaphylactic reactions. Well, this same kind of thing can happen here with mom. So if I give birth to an Rh positive infant, the first time it happens, it may not be a problem, but now the mother has become sensitized, okay, to the Rh factor, and subsequent births can be a problem where that baby may be, if I am born or if I conceive another infant that has Rh positive blood, my immune system may attack that infant, and you can have lots of problems. There are very good medications that can help attenuate this response, and one of the primary medications we use is something called Rogam. Okay? There are also some other types of problems under type 2, and these are drug-induced or drug-associated hemolytic reactions or anemias. And basically what happens here is a drug binds to proteins on red blood cells, and this produces a complex that can cause antibodies to form. And this is actually something known as a hapten. And a hapten is a small molecule that by itself won't elicit an immune response, but when that molecule becomes attached to a larger protein, what we call a hapten carrier adduct is formed, okay? And then that hapten carrier adduct can actually go on to elicit an immune response. And that can happen with some drugs such as penicillins and cephalosporins. These are types of antibiotics, and there are other examples as well, but these medications can bind to proteins on the red blood cells, and then that complex that's formed after the binding occurs can then go on to become antigenic and elicit an antibody producing reaction. Okay, type 3 hypersensitivity reactions are immune complex induced inflammatory reactions. So they're not directly cytotoxic. This is an inflammatory response, and there are many types of disorders that fall under the umbrella of a type 3 hypersensitivity reaction. Problems such as lupus erythematosus, or what we call lupus, rheumatoid arthritis, Goodpasture syndrome, post-strep glomerulonephritis, which is an inflammation of the glomeruli and the nephrons of the kidneys. All of these disorders all fall under the umbrella of type 3. These are immune complex induced inflammatory problems. All right, they're relatively common. All right, these are common autoimmune disorders, particularly lupus and rheumatoid arthritis are relatively common autoimmune disorders where inflammation occurs because the body has created antibodies against its own tissues. The big thing to remember here is it's not direct cytotoxicity. So cells aren't being directly destroyed, but rather there's indirect damage due to chronic inflammation. Okay, type 4 hypersensitivity is known as delayed hypersensitivity. And the classic presentation of this is something that doesn't occur right away, but rather it occurs 2 to 3 days after you are exposed to the antigen. Common example of this is a contact dermatitis, say poison ivy or poison oak exposure. Initially, when you get exposed to these substances, you don't have signs and symptoms until a couple of days after the exposure. That is just a classic type 4 reaction. And that concept of a hapten that I talked about earlier is important here because there is a molecule within poison ivy in particular, okay, uracil in poison ivy, and what happens is this can bind to proteins in the body, and it can produce a reactive antigenic molecule known as quinone. And it is the production of quinone, this hapten carrier adduct, that causes an immune response, and this takes a couple of days to occur. And then type 5 hypersensitivity reactions are very similar to type 2, but there isn't cell destruction necessarily, but there is destruction of certain cell components, if you will, specifically receptors. So you're not destroying the whole cell like you would see here in type 2, but you're destroying a certain component of the cell. Often this is due to destruction of receptors. So some common examples include things like myasthenia gravis. Myasthenia gravis is where the body produces antibodies to acetylcholine receptors, right? So you destroy the nicotinic and muscarinic acetylcholine receptors, specifically the nicotinic acetylcholine receptors in skeletal muscle. And so what this does is this can make it very hard for your patient to have normal movement. They can even go on to have trouble breathing and walking and moving around because of their acetylcholine receptors being destroyed. Pernicious anemia. This is a loss of an intrinsic factor protein, and this intrinsic factor protein actually allows the body to absorb vitamin B12, and vitamin B12 is a very important component when it comes to producing the hemoglobin of the red blood cell. In fact, vitamin B12 deficiency is one of the two most common causes of anemia, folic acid deficiency being the other common cause. So if you have a loss of this intrinsic factor protein because it has been attacked by antibodies, then you won't be able to absorb vitamin B12 and you'll go on to develop anemia. Another type of type 5 hypersensitivity is a type of Graves' disease. This is a type of hyperthyroidism. This is called morbus Graves. And what happens is the body produces antibodies against the thyroid stimulating hormone, or the TSH binding receptor. And normally what happens is, of course, the thyroid hormone is what helps regulate general body metabolism. So what can happen in this case is normally when you are producing adequate amounts of TSH, it will bind to this receptor here, and this receptor will recognize, okay, we've got enough TSH, and a negative feedback loop will occur that will inhibit too much TSH production. But if this receptor were to become compromised through this autoimmune response, then the body won't know to stop producing TSH, and more TSH will be produced, and that will cause a hyper response of the thyroid, and the thyroid will release too much thyroid hormone, and that can lead to this specific type of hyperthyroidism. Okay, I think I'm going to cut it off here. I've thrown a lot of information at you guys in this particular video, but hopefully you found this helpful, and please spend some time studying this and make sure that you are familiar with the general types of hypersensitivity reactions, and make sure that you can identify specific problems within each category of these hypersensitivity reactions. Okay, guys, as always, thanks for hanging in there.